Hi guys, welcome back. This is the nucleic acid portion of our notes, so let's get started. Just reviewing some of the basic information about our nucleic acids. They are made up of the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. The bonds that hold our nucleic acids together are called phosphodiester bonds. The monomers are called nucleotides, and their function is genetic information. Some examples of our nucleic acids includes DNA, RNA, and ATP as well. We're going to look at now how the structure of the nucleic acids affect their function. To do that, we're going to go into detail about the monomer, which are the nucleotides. So our nucleotides are made up of three essential components. Number one, a sugar. And the sugar is going to be made of 5 carbons. And so since it's 5 carbons, it's called a pentose. Number 2, the third component of a nucleotide is going to be a nitrogenous base. And then number 3, the third component of our nucleotides is going to be a phosphate group. So let's look into detail about this monomer before we start looking at how that structure affects the function. So our 5-carbon sugar is a pentose. So that's carbon number 1, carbon number 2, carbon number 3, number 4, and number 5. So there is our 5-carbon sugar. If this carbon, I'm going to put a little star here at carbon number 2. If at carbon number 2 there is oxygen, if oxygen is present, if we draw in a little oxygen right here, then the name of this sugar is called ribose. If at number 2 there is no oxygen present, then we call it deoxyribose. So those are the 2 sugars that we're going to be using in our nucleic acids to make up our monomer, ribose and deoxyribose. And you always look at carbon number 2 to see if that oxygen is present. The second thing on our monomer, the nucleotides, is that nitrogenous base, and the nitrogenous base will always be attached to carbon number 1. We'll go into details about what those nitrogenous bases are, but for now, know that they are at carbon number 1. Last, but certainly not least, is a phosphate group. That phosphate group is going to be attached to carbon number 5. So here we have the basic structure of our monomer of the nucleic acids, which are called nucleotides. A 5-carbon sugar, a nitrogenous base, and a phosphate group. So we're going to take a look at one of the nucleic acids and see how that structure affects the function. And the example we're going to be using is DNA. So remember that DNA, from your 9th grade, you guys learned that DNA was double-stranded. It's the genetic information of the cell. So what I'm going to do is I'm going to draw the 2 strands of DNA coming down. And I'm going to be talking about a little bit about its structure, and then to show you how this DNA actually is double-stranded, I'm going to have to flip the page. So let's start with our sugar, always a 5-carbon sugar. And since this is DNA at carbon number 2, I'm not going to put an oxygen group right there. Here's my nitrogenous base. Here's my 5th carbon, carbon 1, 2, 3, 4, 5, and attached to that 5th carbon is a phosphate group. So there is one monomer, one nucleotide. The next nucleotide I'm going to add is going to be right here attached to carbon number 3. So I'm going to draw a bond to the phosphate group of the next monomer. This is something that many students get confused. When we're talking about attaching two monomers, two nucleotides for this DNA strand, this bond right here is the phosphodiester bond. That one right here. The phosphodiester bond forms between carbon number 3 and the phosphate group of the next nucleotide. So the phosphate group, if this is nucleotide number 1 and this is nucleotide number 2, then that phosphodiester bond would form between carbon number 3 of the first nucleotide and the phosphate group of the second nucleotide. Okay, so to draw my DNA strand and to get the nitrogenous bases to fit correctly, one strand runs anti-parallel to the other. And what that means is I'm going to take my sheet and turn it upside down. When I draw my next strand of DNA, it's going to be upside down with respect to the other one. So here's a nitrogenous base of the first nucleotide of this strand. So there's a nucleotide. I got a 5-carbon sugar, a nitrogenous base, and that phosphate group, and it's upside down compared to this one. The same rules are going to apply. That phosphodiester bond is going to form between this nucleotide and a second one. So I'm going to start at carbon number 3. There's my phosphodiester bond. There's the phosphate group of the second nucleotide. There's my second nucleotide, and there is my phosphodiester bond. Now, between the two strands of DNA, I'll flip this back over, between the two strands of DNA, we are going to find hydrogen bonds right here. And that's very tricky for students to remember sometimes, is the difference between where those phosphodiester bonds and those hydrogen bonds are. So these are hydrogen bonds between the nitrogenous bases of the two anti-parallel strands. My phosphodiester bonds are between carbon number 3 and the phosphate group of a second nucleotide. They always get these two confused. Those hydrogen bonds will form according to what are called Chargaff's rules. So Chargaff's rules for DNA are that adenine is going to pair with thymine. But it's not just a single hydrogen bond that's going to form, it's actually a double hydrogen bond that will form between the A and the T. Chargaff's rules are also mentioning that the proportion of cytosine to guanine is always the same. So cytosine will form triple bonds with guanine. Now, this happens because DNA is double-stranded. Your RNA is single-stranded. And in RNA, when we do complementary base pairing, it's very similar to DNA. The only difference is in RNA, there's no T, we use a U instead. So the nitrogenous bases in RNA are A, U, G, C. If we had to talk about the differences between them, between DNA and RNA, DNA is double-stranded, RNA is single-stranded. The nitrogenous bases in DNA are A, T, G, C. In RNA, they're A, U, G, C. Another thing that I could comment on is that in DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. Thanks, guys, and I'll see y'all next time.